Debbie Russell will not be giving her talk. Chris Lynam will be covering that in his session. He's up first. And so there'll be a little bit of extra time for questions at the end. Like all the other panels, please keep your questions until the end of the talk, and we'll have all our speakers up the front. I'd like to welcome our first speaker, which is Chris Lynam from CEFAS, and he'll be talking about modelling food webs and diversity in the North Sea under multiple pressures.
Okay. Thank you very much. Oh, thank you very much. Okay, yeah, so food web effects due to structures in the North Sea. So this is part of the EcoSTAR project, and Debbie's presented in the past quite a lot of the work that St Andrews have done on seals and other marine mammals. And we at CEFAS have been working with them and doing additional work on benthic interactions and food web modelling, which is where I come in. But today I've got my benthic hat on as well, and I'm going to talk about some of the work that Silvana and Clement have been doing in the project with myself. So we've been doing some benthic trait studies. That's really been Clement's work, looking at functional diversity. I won't touch upon that today because we have a small slot. And together we've been looking at the UK Benthos database in terms of the functional groups that I require in my modelling work for the North Sea. And so we talk a bit more about that. And Zelin from FUICOMS, the other project we're involved in, was really helpful with that line of work. We've had additional data from some of our subcontractors in the Netherlands and Belgium. But that's just a note, really. And then I'll talk a bit about the ecospace development that has really run in from Insight 1, from the COSM project that I had, and is aligned now with some European work that we do in a project called Future Mares, which focuses on climate change and biodiversity. So we're trying to bring the threads together. In this project, EcoSTAR, we still have our what-if scenarios to do with the modelling, and partly it's because I really wanted to hear the updates from all the projects before finalising that, and we've had an extension to the project as well, so we still have time. And that's going to be in collaboration with another couple of EU projects that we're involved with, which focus on cumulative impact assessments, and the modelling that we're doing is kind of useful across all these related areas. So to start off with the benthic work, the first thing that Clement did is he took the UK oil and gas benthic database from the platforms, that's where it's sampled, and looked at the locations of the platforms and then tried to group them into similar environmental clusters. So using additional data on sediments, shear stress, current flow, temperature, salinity, all these sort of basic things, sediment type, that sort of thing. And we've coloured the platforms here based on similar environmental conditions. And then we wanted to analyse the data across all the platforms together, because we're not focusing on an individual platform like Northwest Hutton we've heard quite a lot about. We're just trying to look generally across the North Sea to get some rules of thumb that I could use in the modelling work. And so what Clement really found was there's a little patch here of red. This is a really muddy area. They're very distinct, the platforms in that location. There's another patch up here, up towards Norway, very deep waters. And there's a patch down here off East Anglia, which is much shallower and it's more dynamic. So they're quite distinct. And then in the middle, the sort of the green and the blue is kind of central North Sea, kind of in the middle, really. So we have these 5 different clusters, which we analyse further in terms of these 5 functional groups that we have in the EcoPath, the EcoSIM model that Steve Mackinson and colleagues at CEFAS over many, many years developed. So there are many hundreds of species. It's quite terrifying for an ecosystem modeler. So things are grouped into these 5 functional groups based on similar traits, similar feeding strategies, similar mobility, that sort of thing, and also whether they're large or small individuals. So we looked at the grab data across all the platforms, got the almost 3,000 or slightly more taxa, put them into these functional groups, and then tried to look for changes in biomass, once we move from abundance to biomass, in those functional groups, both near and far from the structures. And in this case, far is further than 250 meters, and near is within 250 meters. And so for these 3 groups that are, the small infauna and the small mobile epifauna are quite well sampled by the grabs. Using all that data together, all the years, everything pulled together, there's, on a log biomass scale here, there was a distinct difference, significant difference in the small mobile epifauna and the small infauna, and weakly with the infaunal macrobenthos too. So closer to the platforms, less biomass, potentially a problem. But we wanted to look at that in further detail. Talking to Zelin, we talked about the contaminants, and then with John Hartley about the temporal aspects of this, which are quite important. So if you look at the contaminants that we saw very briefly earlier in the presentation, across all those platforms, the only thing in common is they all have measurements of total hydrocarbons. There's metals and things, but not all the platforms get sampled. So we did a mixed model here for the 5 different clusters, which is why there are 5 different lines, and looked at how contaminants decay with distance from the platform. And what's interesting is the 2 lines at the bottom, which are the shallow southern North Sea and the very deep area off Norway. The contaminant level was never particularly high there, but it still decays, whereas in the central North Sea, much higher levels which decay. But if we look at it temporally, initially it's not high levels, but they're very high levels at the beginnings of the 1990s, which then fall. And that's because in the 1990s, additional regulations were brought in, and we moved from mud to synthetic oils in the fluids to improve the drilling capacity. So we moved from oil-based to synthetic-based fluids, and then you see that contaminant level comes down. And across all the 5 clusters, we see similar, patterns. And post-2000, we're not really getting the very, very high levels that we had in the 1990s. So there is this temporal pattern. And if I looked across all of those 5 groups grouped together in terms of biomass, near and far from structures, prior to 2000, there was a significant difference, like 35% of the biomass levels near to the platforms as opposed to further away. But after 2000, it's much more comparable and no longer really significant. So overall, those platforms all taken together, there may still be issues in individual ones, but in the broad scale, it becomes much less significant in terms of contamination and in terms of total biomass. But we looked at the 5 functional groups independently, and they correlate in terms of biomass and distance from platform. And so they also correlate in terms of biomass here of small mobile epifauna and contaminants. When there's high contaminants, there's less biomass. But if we look at the 5 clusters separately, we see actually the northern deep area and the southern area, where there were never particularly high levels in the database in the grab samples, in the contaminant samples with the grab samples for the biomass. There was never really significant impact, either pre-2000 or post-2000, when you contrast the reference conditions to the near conditions. But those 2 central ones and the mud patch, where there were high levels of contaminants prior to 2000, there's a significant difference in the total biomass of this small mobile epifauna group. But then after 2000, the values are comparable again. I mean, the sampling jumps around, which is why the levels jump around, and maybe could have standardized that. But there was a clear impact across the North Sea for the platforms for some of these groups where there was contamination. I mean, it's correlative evidence, but it does seem to point in that way prior to those regulations in, I think, 93 and 96, where we moved to the synthetic muds. So in terms of the biomass in the seabed at the platforms, the problem seems to be much lesser now. But we haven't looked at the data in terms of changes in biomass of things attached to the structures and off the seabed, or in larger organisms, larger crabs and lobsters that you don't sample well in the grabs. But this was important information to help set up my model, because working forwards, I don't necessarily need to change the biomass at that location for those functional groups. But I might want also to look at changes further up, which is kind of the review of the information coming from the other projects in terms of changes in attached individuals or bigger individuals. So we're trying to get as much information as we can, whether it be from plankton, mammals, fish, to start working with the spatial model. So we're taking into account now habitats, natural substrates versus the artificial. Natural variation in temperature, salinity, plankton information alongside fishing impacts, and trying to bring that together with the changes in the structures. That's where we're moving. And we're doing that with this EcoBATA, the EcoSIM tool, which we already had a temporal tool, and we developed in COSIM an initial spatial model, and then we've refined it here and working on it further. And this is the list of groups that we have within the model, so from minke whales down to phytoplankton. And it's refined now in that we split the seal group into grey seals, harbour seals in the north and in the south, separate populations, harbour porpoise, and we added in mussels and oysters as separate groups, and we added in kittiwakes as a separate group from the surface feeding seabird group that we had previously. And this mess of lines here is just to show you that this is a food web model, and it's the diet interactions that are really very important in terms of who eats whom. And a lot of these lines are blue, and that means there's lots of weaker linkages. So there's lots of generalists out there. There's a few clear sort of red lines which go from phytoplankton to copepods to sprats. There's more of a food chain, but it's clearly a food web, and there's lots of interactions going on. So we parameterize that with data on stomach contents and on literature review, and then we try and calibrate it through time to time series of data. And this is biomass data. We do the same with catch data, landings and total catches where it's available. So we push the model through time with changes in the climate and changes in fishing effort. We used to use fishing mortality, but we've moved to effort because you can go spatial with that, whereas you can't with an F estimate from a stock assessment. And we have these 9 fishing fleets within the model. So we pushed the model forward from the base in 1990, where we had all the food web interactions, and then we calibrate to try and get close to these time series. But it is not a statistical model. It's kind of more of an iterative process of altering parameters to get closer and closer to the time series. So this is one of the weaknesses of EcoPath EcoSIMs. It's not fully statistical, but it allows you to do lots of things, and it's very, very flexible. So down at the bottom here, we have phytoplankton, which are calibrated against a study from Capuzzo et al., using chlorophyll measurements from the North Sea, and then we have 2 groups of copepods and a less abundant group of carnivorous zooplankton with continuous plankton recorder data from the MBA, and they're doing pretty well. Sand eels are doing quite well, and sprats. There's some temperature information that gives these fluctuations. I think in the main, we're pretty happy with this. Some of the groups are like mackerel, are widely uncertain when we try and do this Monte Carlo analysis to look at uncertainty. And we did quite well with the data that we had from St Andrews for grey seals and harbour seals in the south, but not particularly well with harbour seals in the north. The data's a bit weaker. But when we go spatial, we kind of account for this. And to do that, what we do is we say we've got this calibrated model, we know who eats who, we've got the information on fisheries, and then we bring in additional studies. We did a lot of work on species distribution modeling to get these environmental niches for each of the functional groups, and then that allows you to take the spatial information on the substrates and the environment, along with the preferences of the species and their interactions to give you spatial maps of each individual functional group, which is what we're doing in a nutshell. So we did these improvements for harbour seals, and when we went spatial, we realized that what we really needed to get the sort of match to the time series is we need to inform the model on the changes of the haul-out usage over time, because that's not like marine, it's not a process within the model, but it's clear that the seals have moved haul-outs, and they've moved to the southern North Sea, and we've got more harbour seals off the European coast. So we're trying to get the top predators right in addition to the fishing impacts in this model, and then calibrating against the data as best we can. And the same with grey seals. We've got a lot more of them off East Anglia than we've ever had before, and the data here from St Andrews on grey seal abundance, which we've correlated to with the ecospace output in black. Harbour porpoise is a bit different. They don't have the haul-outs. There's very little change in their biomass over this sort of calibration period. But we've told the model based on papers that actually they're very dependent on sand eels, and they go to sand eel fishing grounds, they're sort of key hotspots for their distribution, which is what a lot of this information is spatially. So we have a lot of information on fish and fisheries, on top predators, and then we can do scenarios. And these are the ones we've done for climate change for future mares, a project that we've just finished the work for, and it's kind of given us new tools and new abilities within EcoSTAR to look at the changes in structures that we need to do. So we did multiple scenarios, and this global sustainability one was a low climate change scenario where we put in lots of hypothetical MPAs, some of them fully closed, no-take zones, and some of them just stopping bottom trawling. And then we reduced the effort to stop too much displacement from those. So these are sort of maps from the 2020s to 2040 to 2080 to 2100 for demersal trawls and pelagic trawls. Demersal trawls are heavily restricted, We also had some restoration going on here for oysters. I don't really want to go into the details of this, but just to show that we can model the change in the fishery and the impact of the fishery, putting in different MPAs or de facto MPAs from platforms, for instance, or wind turbines. We altered bycatch rates and discards and things like that too. And we were trying to find ways to improve the ecosystem, improve top predators, conservation species. And this scenario was one that was useful for doing that. Low climate change scenario was quite important. And so these are increases in biomass that we modeled for all predators combined here. So large fish, like trophic level 4 and up, marine mammals, birds, and sharks. So you can get changes in biodiversity of individual species or different indicators, things like Shannon diversity. This is an example where we've done the scenarios up to 2100 with multiple RCP scenarios, to look at differences in management, differences in terms of protection. So basically, I want to do more of those, look at more of the uncertainty around the climate. But we have the maps of wind turbines and oil and gas from Maria's paper. We have all the seabed habitat, all that other information in addition. And all these projects, and everyone seems interested in these issues at the moment. So we're trying to use this model to address multiple scenarios. And so the last bit is that we have the maps, we can put them in the model, but what do they mean? We can do it in two different ways, which we kind of outlined in the COSM project. We can either say that these locations are just nice habitat and things will colonize within the model itself, or we can do what we did in Future Mars for oysters and say, actually, we're going to put them in. So in this case, if there is production at a location, we can tell the model there'll be extra production there rather than just this is nice habitat to visit. But which groups do I do it for? The work at the beginning on the in-fauna showed that there's less of an impact, in the sediment, but there's clearly going to be things attached, urchins and anemones and whatnot, which will fall into some of these groups. And there's data out there saying that plaice are attracted to wind farms, and you get more crabs and more mud crabs and more oysters and things. So at the moment, I need to define that scenario, and so anyone with any information or information from your projects, I'm trying to grab that while I'm here before we finalize the scenario to make them more sort of realistic. And then we can toy with this, put things in, take things out, and see what the impact is all the way up to the marine mammals. And that's where I will end it.